Welcome back. I hope the coffee break was good. One summer day a few years ago, I received a phone call. I was the chairman of the Swedish Sarcoma Association, and I could distinguish that the guy on the other end was about 40 or 30 or 40. I heard in his voice that he was sick, sad and worried. But what struck me the most in the heart was that I heard the voices and laughter of two small children in the background, playing. The guy who called wondered if there was anything that could be done. He had been told at the hospital that there was nothing more to be done. I normally, of course, we don't do this. We normally don't take part of these discussions as it's a matter for the clinical teams. But I asked him one question: what kind of sarcoma do you have? After all, there's about 150 different types that all require different treatment. You know what he said? At the hospital, they don't know. I have to be clear, it wasn't this hospital, it was another hospital. They have tried some heavy chemo and found out that it had no effect, apart from that he was getting weaker and weaker. I recommended this guy that he should get in touch with Felix Haglund de Flon, an intelligent pathologist at Karolinska Universitetssjukhuset, this hospital. I usually call Felix the medical science answer to Sherlock Holmes. In my more morbid moments, I usually say to my wife that if one day I need an autopsy has to be done on me, it needs to be Felix, otherwise I will scream. Felix looked, it was in the summer, but immediately he looked into the case, and among other things, he recommended that whole genome sequencing should be done, even if it was late in the process. Felix was able to establish that the same genetic variation had appeared 30 years earlier in Fredrik, in a stored sample from a benign tumor that had been surgically removed from his mouth. The analysis also showed there was action that could be taken based on this genetic variation. Felix recommended a chemo treatment, which would, by a high probability, succeed in improving the patient's condition. But Fredrik died before the new treatment could be started. The information came too late. Fredrik was 42 years old when he died and left behind a wife and two daughters, aged 4 and 6. In this seminar, we will talk about whole genome sequencing, what it is, what it can do, and what it can't do, and what it means for us affected by sarcoma, either as patients or as relatives. This Felix isn't, an imaginary friend like kids have. He exists and is here. Felix Haglund de Flon is a part of the panel, and he's a pathologist here at the hospital. And in the panel also we have Roger Wilson. I think you all know him. Founder of SPAGN and a lot of other things. And a professor of oncology, Bernd Kasper from Germany. We'll start with Felix, who will talk about a project that has been done here at the hospital, a quite large project about this, and what it's doing with this and what it means for us. And then we follow up with comments from Bernd and Roger, and we would like to have questions from the audience about it, but short questions. Because the panel, they promised to give short answers. We'll see how this goes, all right? So, Felix, please.
Thank you, Magnus. It's hard to continue after that. So I'm going to talk a bit about what it's like being a pathologist and this study that we started 4 years ago. So you've heard by now that sarcoma is a very heterogeneous and rare disease. I want to start with a picture of Carl Linnaeus, who's a famous Swedish scientist who did his work in the 1730s. And he's often called the father of modern taxonomy. So he built a system how to classify plants and animals, and we still use it today. And it's based on morphology, just like pathology is. So the taxonomy we use today is the WHO classification system, or the Blue Book, or the WHO, as the Americans say. And we use morphology. We're looking at the tumors in the microscope and try to classify them based on how they look. But classifying a tumor is actually much more than just looking in a microscope. So we look at the clinical presentation. Some tumors arise in children, some doesn't. So you have to, a very good pathologist, always look at the patient. For tumors that start in the bone, you also have to look at the radiology or the imaging. So as a pathologist, you also have to have some moonshining as a radiologist. And then we do the grossing, as we say, when we look at the surgical specimen from the patient. And after that, histology comes. Now, this study is about, which we did is about genetics. So that's another layer or a bucket of information that you add into the classification of a tumor. And also, you know, all cells in your body is supposed to have the same DNA. And the way that cells behave are dictated by the epigenetics, the way we modify DNA. So we can also study the epigenetics of tumors to classify them. So in analogy with Carl Linnaeus' work, you can see these two very cute owls, the American and the European barn owl. And morphologically wise, they are identical. They were thought to be the same species. But then someone sequenced these owls. And actually, they were two separate species looking exactly the same. And actually, when they sequenced the species of owls, you can see to the left here that all the red markings are actually new species that were discovered based on genetic information. And similarly to this, here you can see an example of something we call a small blue round cell tumor, because the tumor cells are small, blue, and round. You have a Ewing sarcoma in the upper figure, and you have a newer entity called a sick duck sarcoma. And they look very much alike, but they are clinically very distinct. So sick duck sarcomas don't respond as well to treatments, and they are generally more aggressive. It affects other types of patients as well. So if we look at the taxonomy of sarcomas, we have these WHO classifications, and they are published by a group of experts. And here you can see the publication dates. And one thing that stands out is that the time between these publications is shortening. And early, in 2002, we had 81 entities, and now we have 152 in just 20 years. And I can tell you that that number is just going to increase. So we publish this more and more frequently, and we find more and more entities. And a big thing that happened a few years back was that genetics has always been a foundation of describing new entities. But what's new is that certain entities require or strongly recommend that you have genetic testing to establish the diagnosis. So going back to pathology or morphology, it's about looking at images and pictures. So can anyone here tell me who's the painter of this?
John Constable.
John Constable, very good. You can become a pathologist, sir. However, this is a fake. This is a true John Constable. So there's this fantastic British TV show called Fake or Fortune. It's about, you know, people having these old pictures or paintings, and they want to evaluate who painted this picture. And it's a fantastic show. I can recommend it. But they do all these auxiliary tests. They X-ray the painting. They take samples from the paint and analyze, are the pigment the same as from that time when the painter was active, to establish is it a fake painting or a real painting. But every show ends the same way. To take the painting to a very old person who has spent their entire life looking at paintings from this artist. And that person is the sole person who decides who the painter is, if it's a fake or a fortune, not all these auxiliary things. And that's the role of the pathologist. We are generally very old. I'm 84, by the way. So looking at some images, this is what a sarcoma can look like, and this is of course a fake. This is not a sarcoma, this is a carcinoma. This is a lung cancer that has started to look like a sarcoma in the microscope. This is, again, something that looks like a sarcoma, but it's a melanoma, it's a skin cancer. And here you have something that, again, was called a sarcoma, but this is a nodular fasciitis. It's a reactive lesion. If you just wait, this will probably go away. And you don't want to amputate the leg or the arm of this patient. So we asked ourselves, we had the ability to start to test genetics on all cases. So we wanted to find out what is actually the impact of doing this broad genomic profiling or whole genome sequencing of all the cases that we operate. So what is whole genome sequencing? We basically catalog all the genetic information in the tumor cell. The thing is that to be able to tell because there are variations in the DNA, that's what makes us individuals, right? So we have to have a normal sample to compare with. So it's not enough to only look at the tumor, we also have to look at the normal cell. If I were to print a single genome from a single patient out on regular paper and stack it, it would be 130 meters high. And that's if you read it once. We read it 100 times. And we read the normal DNA 30 times. So that's equivalent of 13 kilometers of paper. It's a library. And what is it that we're looking for? We can look at single letter substitutions within this whole library. But that's generally the kind of genetic alterations that you see in cancer, misspelling. Sarcomas, they have another kind of issue. We have a genomic chaos. So instead of having spelling mistakes, we have disorganization of the genome. So I'm going to summarize. So the study we did starting in 2022 and actually prematurely finished because of the results in 2024, we had 200 patients. And we said that the clinical impact is so important that we need to publish this straight away. And the consequence of that publication was that the hospital leadership actually said that even though this is not reimbursed by the healthcare system, we need to start doing this. So the hospital started to take their own money and just pay for the sequencing. And that was based on that we showed that quite a lot of patients got another diagnosis, and the cost of ineffective care associated with a wrong diagnosis was much, much higher than the actual cost of the sequencing. So now we have done approximately 770 patients since we started. We have, I sat last night and I looked through 685 cases. And approximately 3% of our cases, we had a biopsy that didn't contain enough tumor DNA to do the analysis, so a 3% fail rate, and that's quite decent. And in more than 99% of the cases, we found what we call the tumor driver, so we knew why the tumor, what genetic thing caused this sarcoma. So I'm just going to show you some examples. So this is a presentation of a genome in a very, very simplified way. So here we have your chromosomes 1 through 22. And you can just see that it's a straight line here. That means you have the same amount of DNA as you're supposed to. And then you have the 6 chromosomes here. But you see this area here, you have a lot of extra copies. And this is part of chromosome 12. We call this an amplification. So basically, the genes that are located here, we have extra copies of. And you have an important gene here called MDM2, and there is a gene called CDK4, and they are frequently amplified in certain types of sarcomas, like liposarcomas, or in this case, a low-grade osteosarcoma. So the parosteal osteosarcoma. Here, it's just a confirmation. We knew that this was a parosteal osteosarcoma. So here we can see an example of when the genetics actually changed the diagnosis. This was a bone tumor in the spine of a patient. And looking at the radiology and the histology here, we can say that this looks like an osteoblastoma. That's a locally aggressive but still benign lesion. So you curate it, you remove it, and then you say bye-bye to the patient. When the genetics came back, so this is again just a graphical representation of the whole genome, you can see it's a circle and you have a lot of blue lines. And the blue lines are not supposed to be there. They represent this genomic chaos. We call this chromotripsis. It's thousands of breakpoints throughout the chromosomes and then a shuffling. And this is not something you're supposed to have in an osteoblastoma. So this is the nightmare of all bone pathologists. This is an osteoblastoma-like osteosarcoma. We see it every now and then. So this patient received chemotherapy and another surgery to make sure that the tumor didn't come back. Another example of a patient where we changed the diagnosis was a young male who came with a large tumor in his arm. And this is a classic example of how a sarcoma look like and present. And we made the diagnosis of an undifferentiated sarcoma because that's how it looked like. The genetics came back and told us that this is actually a melanoma, a skin tumor. And that's extremely important because then you can get immunotherapy, which is effective in melanomas. You get a completely different workup. And you start to actually look for the melanoma that spread to his arm. But how often is it that a 25-year-old man presents with melanoma? It's usually not even on your differential. And then I want to show another example, not when we change the diagnosis, but when we find a pattern associated with targeted therapy. So this patient had, again, an undifferentiated sarcoma, which is the most common type of sarcoma. But here we had a deletion of a specific gene that's associated with controlling the spelling mistakes in the genome. And every red dot here that you can see is a mutation. It's a lot of dots. So this tumor is what we call hypermutated. And these mutations are expressed by the tumor cell or the cancer cell. And the thing is that our immune system are super good at identifying things that are strange to our body. So usually these tumors can be identified by the immune system. So we know that if you have this genetics, the tumor needs to turn off the immune system. So in this sarcoma patient, we can actually say that immunotherapy is going to work. That's super, super rare. And this is actually the patient that was the first clinical sequencing in the hospital. This is not something that I've seen this pattern maybe a few times over the last years. So it's extremely rare. So summarizing these 685 patients that we have looked at so far, we found what we call a specific genetic signature in 64%. That's when the genetic signatures tells us what subtype it is. And we don't always need it, but it's super convenient to get it because it confirms your diagnosis. In another 34%, we had a strong support, but not as... I wouldn't be able to tell what subtype it is, but the genetics still support the diagnosis of sarcoma. And then in a minority of cases, the genetics were not helpful. And in a whopping 16% of our cases, the genetics actually ended up reclassifying the tumor. That's a lot. That's every sixth patient. Then I should also mention something that's really big in oncology. That's personalized medicine or targeted therapy or treatment predictive biomarkers. That's different names for the same thing. That's when you have a pattern or a mutation associated with a type of therapy. These might not... So these mutations are shown in clinical trials to somehow improve the efficiency, but they are not silver bullets. It's extremely rare that you get a complete response. But still, for 16% of these patients, we could say that we have strong evidence that supports adding these treatments. And I should also say that in some studies, this number is much, much higher. What we have done is that we have removed the gastrointestinal stromal cell tumors because 100% of them have these mutations. So we want to be real here and say it's the high-grade sarcomas that's the real problem, and we don't want to pump up the number too much. There were a minority of patients that were diagnosed with sarcomas that with the help of genetics, we could actually tell that they were benign. It's about 1 or 2% of the patients. But that's a huge impact going from that you have a malignant tumor to you have a benign tumor. And then I should also say that if you read these ESMO recommendations, they say that generally genetics have no help if you have one of these pleomorphic or undifferentiated sarcomas. But what we have started to see is that if you do whole genome sequencing, you're able to identify that a significant subset of these are actually melanomas. And they show no trace of melanoma markers. The only way of identifying them is doing a whole genome sequencing. And I think that might be one of the new things that we will present and actually try to make, to spread that, because this is such an important group to identify, because for melanomas we have a much more effective treatment. And then we have, I won't show you the data, but we have made health economics showing that it's actually feasible to do this. And we're not alone. There are other groups who have done whole genome sequencing. And I would say that the numbers here fluctuate a little bit, depending on which patient you choose to include. I think we include many, many more patients. We have a wide inclusion criteria. We sequence any case that is suspicious for sarcoma, not only sarcomas, but anything that is believed to be a sarcoma. But I think that the number stands. One other thing which I think is important to mention during this study, I had elder colleagues who were a bit skeptical, who said that we don't need this technology, we are good enough. But during the study, as we started to get the genomic data, it started to knock on my door every now and again. And my colleagues, the senior colleagues, they came by and asked, has the sequencing arrived yet? I don't want to sign out this case without knowing the genetics. And I think that really speaks for itself, that that people who were critical actually adopted this method happily after a while. And one other thing I should mention is that the common way of doing this in other hospitals is that you have a pathology department and a genetics department. And the pathology and genetics, they work independently, and then you have a genetics report coming independently from the pathology report. And I think many pathologists get a bit frustrated over that, because someone else is influencing the diagnostic and classification. So what we do is that we have a fully integrated pathology report. So it's the pathologist who actually signs out the genetics report and say this is how it influences the classification of the tumor. And I think that very few places actually does that. This is not a one-man show, that there are so many people to thank, all the patients and the hospital and the research institute and all the groups and of course the funders of this study. So with that, I'm gonna close and give the word back to Magnus.
Wow, good one.
Thank you, Felix. At once after this study, the hospital decided that this should be clinical standard for all suspected sarcoma. Why was that? What was the reasons?
Yeah, I think I should, actually applaud the leadership, because they made this daring decision. I think in nowadays everything is about economics, but I think that they looked at the data and said we are a university hospital, we need to lead and create new knowledge and set a standard. So they dared to take that decision, and I'm very happy and proud that they did that.
Did something in the results surprise you? Was it a surprise for you in the study?
Well, yeah, lots of surprises. I mean, because you don't expect to get your diagnosis changed, right? And some old pathologists told me that you should only work at a sarcoma center for 5 years, because after 5 years, that's when your mistakes come back. We have a good sense of humor. But the nice thing about this is you get the feedback at the same. You said, okay, I think it's this, and then a few days later you get the genomics report, and then you go, oh, I'm wrong. And that kind of instant feedback is so good to train you to become a better morphologist. So then you see straight away, you go back to the case and you question, okay, how should I approach this kind of case the next time? And then I should also say that in our original study, the first 200 patients, the number of change diagnoses was 7%, and now we're up to 17%. And the reason is that we have become better geneticists. We have gotten all this information we have gotten new tools to help us classify the cases. So we have, I think that even though the number of reclassifications have gone up, it's also a reflection of that we have become better. Yeah.
Ben, from a clinical perspective, are you surprised, or was it?
I mean, to be honest, I'm not really surprised. We know that there is something like almost 20% of misdiagnosis. But the nice thing about that study is that it is really proven here. And I think when talking about all these NGS stuff, we always have to differentiate between two implications. There's the diagnostic implication, and Felix actually focused on that very much in his presentation. And on the other hand, there is the therapeutic implication, which I think is much more difficult. And for the diagnostic implication, I think the data is great, and it is so meaningful for the patients if the diagnosis really changes. I mean, when talking about you showed this heterogeneous disease with more than 150 subtypes, and I mean it makes a difference if you have a liposarcoma, or if you have whatever another rare subtype of sarcoma. It makes a difference if you do a surgery or if you do a specific chemotherapy or not. So this is impactful for the patient, and it's even more impactful for the patient if the diagnosis changes from malignant to benign. And this is also even the case in almost 2%, I think. And I mean, imagine you are faced with a diagnosis of a malignant tumor, and 3 weeks later you are told, oh, it's benign, we just have to take it out, and then it's done. And so you can imagine what you are going through in these 3 weeks. And so yes, the high rate of misdiagnosis, 16-17%, is actually really the most appealing about that study, I think, and this is really impactful. And I think in this respect, Angie is a perfect tool to help the pathologist adding on the conventional pathology.
Roger, what do you say? Are you surprised?
I wasn't, in one big respect, because at Cambridge, and it was mentioned on Felix's slides, Cambridge, they've been running a similar study. It wasn't as large scale, and the medical oncologist who worked on that study is actually someone I know quite well, and she had been talking to me about the work that they were doing, just hinting at there were going to be some interesting results when they published, which there were, along very similar lines to what Felix has presented, although the study was much smaller. It was only 67 patients. The one thing that struck me about the Cambridge results was that they were able to highlight one particular patient, who was a patient who, I think it was a pleomorphic sarcoma, but he had lung metastases, and they were not treatable under standard of care, and he had a 4-month, maybe 6-month prognosis. And yet when they whole genome sequenced him, they spotted that he might be suitable for a checkpoint inhibitor. Now, no checkpoint inhibitors are approved for use in sarcoma at the moment. They sought exceptional treatment approval and funding, and got it. And 3.5 years later, that man was still alive. In fact, he was of Indian origin, and he'd been home to India to visit family and friends, and in a perfectly normal way. So that was the thing that surprised me in the context of whole genome sequencing, that you could arrive at a result like that by using this approach. And when Felix went through his results very briefly with us online a couple of weeks ago, I got a very clear admiration in my mind for the management at the Karolinska here, for taking that step forward to whole genome sequence every patient, because we asked for that in the UK on the basis of the Cambridge results. We were refused by the National Health Service because they were scaling the demand for whole genome sequencing in the whole of the cancer oncology treatment domain. And there were so many requests on a volume basis per annum that they had to cut back on virtually all of them. The only one which got everything approved was in pediatric cancers, including sarcoma, I hasten to add. So adult sarcomas, it's a case-by-case example for funding by the NHS as a national organisation. And I just compare that with the decision your hospital here made, which just seems to me rather sad. But that's the way things are. And if you, as I did, question why a decision was made that way, it was done on the basis of looking at the availability of whole genome sequencing in the academic arena. They didn't look at commercial providers, but only within the academic arena, which would be paid for through the state. And they looked at demand, and they had to do a balancing exercise, and that's what came out. So surprise is one thing, but the logical sequence of events that followed it leaves me with a sense of admiration for Karolinska and hope only for the future as far as the UK is concerned.
Bernd, what do you say from a clinical perspective? Should this be clinical standard?
I think it's more and more becoming clinical standard, I would say. Let's put it like that. And but the problem is and that's always why I would like to differentiate that. For the diagnostic side, I think it's very clear. It can certainly help to either ensure the diagnosis or come up with another diagnosis. I think it's getting much more complex if it goes to the therapeutic side, because the problem is if you do it in every patient, you are generating a lot of hope for things that might not arise. And I mean, the problem is the percentage in which you actually find what we call a targetable mutation. That means a mutation which we can target with a drug or with a whatever, and with a kind of therapy. The percentage of that is very low. And I mean, even if you find it, the problem is the drug is not available, the drug is not approved in your country, or the drug is not approved for this indication in your country, or the drug is pretty much expensive, or there is no clinical trial available where you can put the patient in. So this is a bit the problem. And having said that, you have to, we still have to answer a lot of questions. And these questions are, when actually should we do NGS? In which situation? At primary diagnosis, when there are metastases, in every patient. I mean, all these questions are still not answered. And I think we have to answer them from all these perspectives, also taking into account the patient perspective, because at the end, the problem is that we very often cannot fulfill the promises we are making at the very beginning. And I think we have to be careful here. But yes, taking together, it is definitely something which I think will more and more become routine practice if we have enough money. I mean, that's another issue, of course. A lot of us are working in the academic field where money is always limited. And I mean, you are often obviously very fortunate here to do it in that way. But in Germany or in many other countries, we are far away from doing it in every patient still. And yeah.
But is it a cost, Felix?
Well, I have to agree with the issues with this treatment predictive biomarkers. I'm the first to really support that. It's going to be a very few patients who have effective treatment from this today. And so what we have done is now we have a catalog of all our whole genomes, and we have that accessible to the phase 1 unit. So if there is a new clinical trial coming, they can basically open the catalog and see how many patients do we have at Karolinska who has this target if a pharmaceutical company want to open a trial. I think that's a good thing. And with cost, I think that we've seen that the cost of doing sequencing is getting lower and lower. I think we're down to 1,000 euros for a whole genome. It's in a few years going to be 100 euros for a genome. But the cost is not only the sequencing, it's the data management, computational things, and then the time for healthcare personnel to interpret the data. For me, I can actually, one of these cases I've shown, I rarely spend more than 5 or 10 minutes per case. But that's on the diagnostic side. But I think here that we will get stronger and stronger tools that helps us. And I think if I'm to speculate now, in 20 years, I think we're going to have these huge catalogs of whole genome cases. And basically we're going to have AI-trained image analysis on these regular histology slides that can identify genomic patterns just from the histology. So maybe there might be, I think we will still sequence, but in 10 or 20 years we will probably do an AI test followed by a confirmation on genetics. But who knows?
Roger, how do we communicate this to the patients? How do the doctors communicate this to the patients so we won't get our hopes up? Obviously, everybody won't get cured just because of this. I'm sorry to say.
Yes, I think we're moving into an evolving area, aren't we? Because it's going to shift fairly quickly, I suspect. And I'm delighted to hear Bernd saying that he feels it's going to become part of the, if you like, the standard package of support for diagnostics, even if there are concerns about how we turn that into therapeutic information. Getting those two distinctions, or that one initial distinction over, is, I think, part of the key to how we communicate it. Because diagnostics is undoubtedly, or accurate diagnosis, is undoubtedly the thing that's in the forefront of our mind in the whole of the sarcoma arena. Getting there quickly, getting there accurately, and turning that into the effective first-line treatment, which is hopefully curative in most cases, is our challenge. And that would suggest that we've got to rely on histology in the immediate term, with support from sequencing in individual cases as determined by the diagnostic clinician. Now, many of those diagnostic clinicians are surgeons, and they will rely very strongly on their pathology colleagues. Fewer of them are oncologists who are perhaps more used to the scientific bench, although I would hesitate to make that as an overall general rule. So I think the message that we need to construct from the patient viewpoint in terms of communicating the value and the power that lies in whole genome sequencing is to say that if it's available and if you are asked as a patient to have your tissue sequenced, do so. By all means ask, but if the diagnosing doctor says no, it's not going to offer us anything of value, you listen to them and accept that in the first instance, because you will realize from that if you actually asked for sequencing and sought sequencing, you would probably have to ask a pathologist from another hospital and a diagnostic lead from another hospital to actually take over your case. And the answers may be just the same from them. So in the first instance, respond if sequencing is suggested. Don't push it if it's not. However, if the first line of treatment is not successful, if there is, for example, a fairly immediate localized recurrence, or if there is the need for medical oncology to support a curative pathway, by all means ask for sequencing, insist on it, because it may well be that the answer that the oncologist is seeking is not there from the pathological information he's already got. So that's somehow we put that together as a package of information. I'm not entirely sure, but that's the kind of message I think we need to get across.
Yes. Let's go over to questions from the audience. Short questions, short answers, and I would like you...
Sorry.
No, it wasn't. I wasn't picking on you. Short questions, short answers, and please say who you are and where you come from. There, up there.
Hello. It works. Okay, it's Andy from Ewings again. So you've talked about diagnostics and you've talked about a therapeutic pathway, but is there another aspect to this, and maybe this is an ethical one, and that's prognostic capability? And I hesitate when I'm talking to pathologists and oncologists, because I'm neither. But for instance, in Ewing sarcoma, if you've got a SPAGN mutation, then that gives a poor prognosis. We know this already. I'm not sure if a SPAGN mutation is something that's born out of whole genome sequencing or some other method. But if you find you've got it, then your prognosis is typically poor, about 20% of cases. So putting aside diagnosis and putting aside whether there is a therapeutic pathway, having found that aberration, is there not an ethical question about, we've found this, your prognosis may be better or poorer, and as a patient you should have the ability to be given that information?
Ben.
I mean, that's a very good question, and I think there is actually no answer to that. And that is always the problem, that these methods may offer us information which we sometimes maybe don't want to know, and or even don't want to give to the patient. And yes, I mean, what should I say? That is a problem, definitely.
Mm. Mm. Well, question up there.
Thank you. Nick Puntis, Just Cancer UK. Roger, I'd like to give you a bit of hope on what you were saying about the UK. And this may resonate with other governments, not being political, around the world. So in the UK, we've just had the Rare Cancers Bill, has just been enacted in Parliament. It's now the Rare Cancers Act. As what I would call a very nice coincidence, there's also just been the production of the new 10-year cancer plan at the same time. The 10-year cancer plan now has a whole chapter about rare and less common cancers. And the other charity, as you know, that I'm involved with is Cancer 52, that represents all rare and less common cancers. We've been heavily involved in both of those. And genome sequencing has been one of the areas that we have really pushed for. So watch this space, because I think it means that the government are now forced to put more money, more resource into these things that are going to make a big difference. So not necessarily a question, but it might generate questions about other countries in the world and their wanting to get to the same situation you are here.
Well done, really. Well done. Comments?
Thank you, Nick.
Let's see. Up there.
Hi, my name is Sam Hackett, and I'm one of the Sarcoma UK nurses. It's a pathology question. as we've just mentioned, we had whole genome sequencing running in the UK and it got pulled except for paediatric and TYA patients recently, which was heartbreaking. And we rely on panel testing. And I just wondered, when you think about your research, how many of those would you have got the result if you also only could rely on panel?
That's a very good question. So a panel sequencing is when you don't look at the whole genome, but you focus on the 0.1% of the genome that's cancer associated. And generally, panels are designed for carcinomas, not for sarcomas. So in cancer in general, panels are great, but I think you have a high risk of missing out on quite a lot of things if you use a panel that's designed for lung cancer or whatever. So it's all up to if you have like a panel designed for sarcoma. If you have a sarcoma panel, it might be quite good. So yeah.
Nicholas.
But I should say that the majority of commercial panels are not sarcoma panels. So we have been running a commercial panel here before we got whole genome, and I have had so many cases that the panel comes back blank, saying we don't find any aberrations, and that's just, you don't, you're looking for the wrong things.
Nicholas.
I'm Nicholas, pediatric oncologist. Sorry, I have one question, but I also have two short comments. I was very intrigued about STAC2, TP53 mutations and Ewing. Of course, we're always frightened by if we find that, because we do whole genome on all our pediatric patients as well. But I mean, the one patient I'm thinking of where I think, oh, this is not going to be good. There was nothing I told the family. He's still alive 3 years later without any issues. So I mean, statistics is one thing, but the individual patient is another. And then the question is, I think, therapeutical relevance. The question here, I think, is what percentage do we need to justify doing that? To me, I want to know about these 2 or 3% of patients that harbor an Amtrak or ALK fusion, because that makes a difference. I also want to know one out of 100 patients that has a hyper mutation phenotype, and we have those patients that we then can give, rather than palliative care, we can offer immunotherapy. Also have those patients in mind that are alive 3 years after everyone has given up on them. So I think it really boils down. What is the number needed to sequence that we satisfy ourselves? Of course, you can look at it from, okay, for 95, 97, I don't know where the cutoff is. It won't make a difference. But to me, as a pediatric oncologist, there's 3, 4, 5%. This is enough. And then what we did not touch upon at all is pathology is expensive the way it is. And sequencing is one part of it. If you do a commercial panel, and that comes at similar costs. If you then need to run additional tests, that also adds up in the health economical analysis. My question that I had, though, is now we're talking about whole genome and whole transcriptome sequencing, and the methodology is evolving. So, Felix, or the panel of you, what do you think when we start doing single-cell whole genome sequencing? Does this bear the potential of getting another quantum leap, or will this just not be necessary to even consider this outside of a research setting?
I think, I think we will push single cell within 2 years here, but not only because of the information, because single cell sequencing is that instead of sequencing thousands or millions of cells at the same time, you're sequencing single cells. And what that does is that you can see how different tumor cells are different. That's gonna, if we're talking a lot of data now, that's gonna be very hard to fathom. But I think that's just technological evolution. But maybe we have to, the way we think about how this information affects our treatment plan have to change, because there is no way you can use that resolution. So historically, how we've done is that we have categorized tumors and say, okay, this tumor gets this treatment and this tumor gets this treatment. But you're going to have so many factors, so it's going to be very hard to make this kind of categories. So I think the way we stratify patients for treatments has to change with that kind of information. But one thing I realized during these years we have done is that it's very nice to work with something when you're curious about it. And 10 years ago, when I didn't have sequencing, I looked at these cases, I read lots of literature, I tried to figure out the tumor, and I reported that, and that was the end of it. And I had this like gnarly feeling that I couldn't understand the disease at its core, right? But with sequencing, I go home every day and I feel that these cases I signed out and these reports I made, I really, in the majority of cases, I really understand the disease. And I feel very good about delivering that information, that it's accurate. And that, because my work is about understanding the disease, not the patients. But and it's less complex to just understand the disease than the patient. But that, I want to say that it's very nice to be able to be curious about your work, and that it's been extremely helpful, this super powerful tool in understanding the disease.
And I actually very much like your discussion about a possible threshold. I mean, actually we cannot give a possible threshold. That's the problem. And NTRK is such a nice example for that. I mean, if you find an NTRK mutation, then you have a perfect therapy to give. And but to be honest, I have not found a single one in my whole career yet. Maybe I'm not old enough, or maybe I haven't looked enough. I don't know. But yeah.
Three in the next year.
Yes. But yes, I mean, that's a perfect discussion, and hopefully this discussion will not be answered by the regulators or somebody like that. And but yes, I mean, for a single patient, finding a targeted mutation is, of course, a big success.
Thank you to the panel. I have to tell the story about how we started this, how you started this. Felix was calling me in the summer. I was sitting outside with a drink, I have to admit, and he was calling and said, Magnus, I can apply for a project with whole genome sequencing, and it's huge. And I said, that's great. When do they need this application? And they said, by the weekend. And I said, Felix, it's Thursday afternoon now. It's no problem. It's no problem. I'll do it tonight. And during the night, he was mailing out to all his colleagues, Sarcoma colleagues, asking, I know you all have a lot to do. I know you have massive things to do, and this will be really heavy. Are you all on? Are you all in? And they said, yes, sure. What a fantastic thing. And that's, it's great being here at the hospital with that attitude. Thank you.
The truth is actually worse. So I came home with my first son from the maternity ward in the evening, and I got the phone call. And the deadline was not the weekend, it was the same night. So I had 4 hours to write the proposal.
Oh, well, even better.
Yes.
Thank you very much, Ben. Thank you, Roger, and thank you, Felix. We have